Welcome to the About the Face podcast, sponsored by the American Academy of Facial Plastic and Reconstructive Surgery through the Facial Plastic Surgery and Aesthetic Medicine Journal.
Hello, my name is Rémi Hervochon. I'm an ENT surgeon from Pitié-Salpêtrière Hospital in Paris, France. Let me introduce our study dealing with cerebral plasticity after lengthening temporalis myoplasty in facial palsy, which is a magnetoencephalography study. Lengthening temporalis myoplasty, which is also called orthodromic temporalis tendon transfer, is a surgery performed on patients with facial palsy. It consists in suturing the temporal muscle to the peribuccal region. It allows a reanimated smile by contracting a muscle whose initial function was mastication. This suggests cerebral plasticity. Magnetoencephalography is a functional brain imaging method allowing non-invasive detection of magnetic fields produced by electrical activity of neuronal ensembles. We performed a pilot study with magnetoencephalography to map the somatotopy of the face in controls and in patients after lengthening temporalis myoplasty during simple motor tasks. Our main objective was to obtain preliminary evidence for the face motor cortex reorganization after this surgery of lengthening temporalis myoplasty. We included 11 controls and 8 patients who underwent the surgery and physiotherapy. Participants performed 5 motor tasks with magnetoencephalography and electromyographic recording. They were thumb flexion, eye closure, smile without beating, tongue protraction, and mastication. We localized the maximal brain activity sources within the precentral gyrus and measured the L distance between this point and the vertex. We performed an ANOVA to test between group comparison for each task and between task comparison within each group. A student t-test and a TOAST, which is a 2 one-side test, were performed on L distance between tasks. We also calculated a somatotopy index. This index is equal to 1 in the case where cerebral sources are found in the exact normal order of the Penfield homunculus. On the other hand, this index is different from 1 in other cases. In controls, we found the exact same order than the Penfield homunculus, which is from the top to the bottom, the end, eyelid muscles, smile, tongue, and mastication. For smile and mastication, there was a statistically significant difference in controls, but no such difference was observed in patients after surgery. In the control group, 82% had the somatotopy index equal to 1, which corresponds to a normal order, the same of Penfield homunculus. On the other hand, in the post-surgery group, only 25% of patients reached the normal order. To conclude, in controls, the cortical motor areas were organized according to Penfield's motor homunculus. After the surgery, the organization was different. Smile and mastication sources were found to be not statistically different and seem to overlap. A limitation is the absence of preoperative data, raising the possibility that the changes that we observed were not solely due to the surgery. That's why we are leading further research with larger sample size and longitudinal data to fully characterize this brain plasticity. Thank you for your attention.
We'd like to thank the authors for their commentary and thank you for listening to our podcast. If you'd like to explore more, please feel free to check out the article in print at the Facial Plastic Surgery and Aesthetic Medicine Journal or online. We'll catch you on the next one.